Thank you. I'm Lizzie Smyth. I'm a medical oncologist and a clinical researcher at the Royal Marsden Hospital in London. We currently have a trial open, which we're screening FGFR translocated cholangiocarcinoma patients for. So what I'm going to do is give a really broad overview of the FGFR pathway in cholangiocarcinoma, the epidemiology, the biology, and the treatment. And hopefully that will provide a nice introduction to the third speaker, Lipika's talk. These are my disclosures. So the fibroblast growth factor receptor is a transmembrane receptor tyrosine kinase. Am I eating the microphone? Thank you. That's better, yeah, thank you. That's involved in very many cellular processes, including growth, proliferation, differentiation, and in survival. The FGFR is composed of two domains. Extracellular is comprised of three immunoglobulin-like chains. The intracellular component is a single split tyrosine kinase domain. The ligand, FGF, binds to the receptor. And this interaction is stabilized by heparin sulfate proteoglycans. There are 4 described fibroblast growth factor receptors, and they exist in many different isoforms. So there are many more ligands than receptors. There are up to 22 FGF ligands described. So what happens? When ligand binds to the FGF receptor, it causes receptor dimerization. 2 parts of the receptor join together, and this leads to autophosphorylation of intracellular tyrosine kinases. Those go on to activate many different intracellular signaling pathways. You've seen this diagram today. As Dr. Zhu said, it is very complex. The main pathways that are activated are MAP kinase, PI3 kinase AKT, and sometimes PLC gamma and STAT pathways. It's important to remember that the pathways which are activated are context dependent on the receptor that's activated and sometimes the cancer in which that receptor is activated. So because FGFR is involved in so many important cellular processes, it's very frequently dysregulated in cancer. This might be by direct effects of direct genomic effects, like mutation, amplification, or fusion, or other effects such as alterations in paracrine or autocrine signaling, or effects on angiogenesis. So the FGFR receptor, or the FGFR gene, is one of the most frequently mutated in cancer. We see mutations in FGFR3 in about 80% of low-grade bladder cancers, 14% of FGFR2 mutations in endometrial cancer. Amplification, or extra copies of the FGFR gene, are a validated oncogenic driver in squamous lung cancer, breast cancer, gastric cancer. FGFR2 amplification is present in about 9% of gastric cancers, and we've seen some encouraging early results in highly amplified gastric cancers with FGFR inhibition. FGFR4 overexpression is associated with an epithelial mesenchymal transition phenotype in cholangiocarcinoma and is associated with adverse prognosis in those patients. But really what I want to focus on today is FGFR fusions. So fusion is when 2 genes, which are previously independent, are joined to each other. This can occur because of a translocation, an inversion, or a deletion. And what frequently happens when these 2 genes join together is that the protein adopts a formation that allows it to homodimerize and activate intracellular signaling in the absence of ligand. So there's been a lot of work on FGFR2 fusions in the past number of years. And we've seen those at low levels in CNS tumors and bladder cancers. But by far the most prevalent, at least 15%, is intrahepatic cholangiocarcinoma. So I'll just talk a little bit about the identification and the epidemiology of FGFR fusions in cholangiocarcinoma. So the very first, among the first FGFR fusions which were described in cancer was by this group from University of Michigan, and 4 years ago in cancer discovery, using deep sequencing and paired-end RNA-seq, they described 4 index cases of FGFR fusions, and 2 of those were cholangiocarcinoma. They're on the right. The first of those was a 34-year-old female with a cholangiocarcinoma. Her FGFR2 was fused to BIC1. The second was a 62-year-old gentleman with cholangiocarcinoma. Again, his FGFR2 gene was fused to BIC1. Now, the conformation of an FGFR2 BIC1 protein means that it frequently homodimerizes, joins to itself and activates intracellular signaling of the FGFR pathway, leading to cell growth. So following identification of the 4 index cases, those investigators went on to evaluate other sources of RNA data, including the Human Cancer Genome Atlas. And what they found was 24 cases total of FGFR fusion cancers, scattered across FGFR receptors and cancers, but including cholangiocarcinoma. And most of the fusion partners carry these dimerization motifs, which mean that they're more likely to homodimerize and activate intracellular signaling in the absence of ligand. So this suggests that FGFR fusions may be an oncogenic driver in multiple cancers. So following the first description of FGFR fusions in 2013, there's really been a real expansion of research in this area. Among the first to describe the prevalence of FGFR fusions in a Japanese data set, these investigators sequenced 106 cholangiocarcinoma, intrahepatic and extrahepatic, and they found 9 FGFR2 fusions for a prevalence of 14%, so roughly 1 in 6 patients. They described the FGFR2 BIC1 fusion, which was previously described, but also a novel fusion. In this data set, FGFR fusions were mutually exclusive with KRAS and BRAF mutations, and they didn't find any difference between age or gender in patients who were fusion positive or negative. And they also suggested that patients who were fusion positive had comparable survival to patients who were fusion negative, although probably with 9 patients it may be a little underpowered for that analysis. Also, they found that patients in this data set with FGFR fusions were more likely to have been diagnosed with hepatitis B or C in the past. And about the same time, these researchers from Mayo Clinic established what the prevalence of FGFR fusions in a US data set was. They used a different methodology. This is brick apart fish. And what they found on a data set of about 100 patients was again a prevalence of 13% of fusions. Different from the Japanese data, they found that patients with fusions were more likely to be female. They were also more likely to be younger than patients without fusions. And they also established that patients with FGFR fusions had a much longer median cancer-specific survival than those without. So these are different results than from the Japanese data set, and it may reflect differences in disease etiology or biology in different regions of the world. So through 2014, 2015, there was an ongoing stream of publications discussing FGFR2 and cholangiocarcinoma. And in each of these previously described fusion partners were identified, but also novel partners, and I've highlighted those in red. And really the proportion of FGFR rearranged tumors is dependent on the series. The series that identified the highest proportion of FGFR rearranged tumors in cholangiocarcinoma was this one. And the patients in this series come from Spain, Italy, and the US. They identified a prevalence of 38% FGFR2 BIC fusions, but also a novel fusion of FGFR2 PPH-LN1 in 16% of patients. And interestingly, the novel fusion and the FGFR2 BIC fusion were not mutually exclusive. And that's a little surprising because it's difficult to understand how that might confer a selective advantage on those cancers. However, they didn't perform single-cell analysis, so it may just represent distinct subclonal drivers. Interestingly, in this data set, and again, counter to what we've seen in the Japanese data set, there was a significant correlation between KRAS mutation status and FGFR fusions. So last, probably the biggest data set that's evaluated FGFR dysregulation, now not just fusions, in the US, was published by Dr. Jane, presented by Dr. Jane at ASCO this year. And she evaluated almost 400 biliary tract cancers and found that a large proportion, 95, had abnormalities of FGFR signaling, the vast majority of these FGFR2. And of these 95 patients, 87% were intrahepatic cholangiocarcinoma, again, a preponderance of female patients, and again, a fair proportion under the age of 40. And of these, 59 were FGFR2 fusions in intrahepatic cholangiocarcinoma. So what does the presence of these, what does this mean clinically for the patients? So they performed an analysis of overall survival in patients who were FGFR dysregulated or not. And what they appeared to demonstrate was that patients who had FGFR dysregulation had a longer median overall survival than those who did not, 36 versus 22 months. Importantly, this analysis was performed in a population who did not receive FGFR-directed therapy. So this suggests that patients with FGFR dysregulated cholangiocarcinoma or biliary tract cancer may have a slightly more indolent biology than those who do not. So moving on to the biology and the treatment of FGFR rearranged cholangio. So the presence of a genetic alteration on its own doesn't prove that it's a driver. So there's been a lot of work to evaluate the effects of FGFR fusion in cancer over the past couple of years. In vitro data supports the fact that FGFR fusions cause receptor hormonal dimerization. And these results demonstrate activation of the FGFR pathway in cells which have FGFR fusions, here showing high levels of phospho-FGFR and phospho-MAP kinase, indicating that these fusions do cause activation of the signaling pathway. And importantly, FGFR fusions cause oncogenic transformation in vitro and in vivo. When we take fibroblast cells and inject them with a retrovirus that contains an FGFR2 fusion with or without an active kinase domain, you can see on the left those with an active kinase domain show increased anchorage-independent growth, so they grow tumor balls. And if we inject those cells into the flank of an immunodeficient mouse, they will grow tumors. So this demonstrates that the FGFR fusions have act as oncogenic drivers in these cells and have a transforming capacity. Importantly, the fusions are also sensitive to FGFR inhibition. Across multiple models of FGFR fusion cell lines, use of small molecule inhibitors of FGFR cause a reduction in FGFR signaling, here demonstrated by decreases in phospho-FGFR and phospho-MAP kinase. And also, this subsequently causes a reduction in cell growth, in tumor growth. So therefore, by blocking the FGFR pathway and reducing tumor growth, we're proving that these FGFR2 fusion cells are addicted to FGFR signaling. And so this would provide a clinical rationale to proceed with FGFR-directed therapy for patients who have FGFR fusion-positive tumors. So, because of the large number of FGFR fusion or FGFR dysregulated tumors, which have been described across tumor types, there are many different FGFR inhibitors in development, and this list is not exhaustive. The first very encouraging results for suggesting that FGFR fusion-positive cholangiocarcinoma patients might benefit from FGFR-directed therapy were with the relatively unselective FGFR inhibitors, davitinib and ponatinib. But since then, several selective inhibitors have been developed, including BJG398, ARQ-087, AZD4547, the Insight compound. Also, we have some irreversible covalent inhibitors of FGFR in early phase development, TAS120 and PRN137. And then also in early phase development, there's an isoform-specific monoclonal antibody, which has had some interesting results in gastric cancer, and some FGF ligands, ligand traps, also in early phase development. So what I would like to focus on a little bit today is BJG398 and ARQ-087, as they've had some very nice results, some of which you've seen already in FGFR fusion-positive cholangiocarcinoma. So BJG398 is an orally available, very potent inhibitor of FGFR1, 2, and 3. And the structure of BJG398, a complex with FGFR, is shown on the left. In kinase assays, it's a really potent inhibitor of FGFR1 to 3 at nanomolar quantities. And importantly, in an assay against many different kinases, it's very specific. So the closest kinase that it inhibits is FGFR2 or VEGFR2, which has very strong sequence homology with FGFR, and it inhibits it at 70 to 100 times the concentration of FGFR1 to 3, which suggests that it's a very specific inhibitor and should not have too many off-target effects for patients. So in preclinical testing, BJG398 inhibited the growth of cell lines that have high levels of FGFR2 or NA, but did not inhibit those that did not, here on the left-hand side of the graph. And also on the right-hand side of the page, it inhibits the growth of tumors in mice, which are FGFR2 amplified. So therefore, there was a very good basis for moving forward with this drug in FGFR dysregulated tumors. And I'd like to thank Dr. Ajavle for providing me with some of the slides discussing the BJG398 study. So this, I'm sure you're aware of this study, presented at GI ASCO 2016. So this was an international phase II non-randomized trial, BJG398, in patients with advanced or metastatic cholangiocarcinoma. They were required to have an FGFR fusion or other genetic alteration in FGFR, and they must have progressed following prior cytotoxic chemotherapy. That cytotoxic chemotherapy being standard of care, gemcitabine or gemcitabine cisplatin. They were treated with BJG398, 125 milligrams once daily on a 3 weeks on, 1 week off schedule, and they were treated until progression or unacceptable toxicity. So on the study, 47 patients were treated. Most patients had had more than 2 lines of therapy, and more than 10% had had 4 lines of therapy. So this was a heavily pre-treated patient population. The vast majority of patients had FGFR2 fusions, and we saw Dr. Zhu present earlier what a variety of fusions were present in these patients, what a variety of fusion partners were present. And 16% had other abnormalities of FGFR signal, including mutation or amplification of FGFR2 or 3. And so this was the first take-home message from the study. This is the waterfall plot demonstrating responses to BJG398. The response rate was 14%, and stable disease rate was 58% for a disease control rate of 72%. But what's really nice on this graph is that for the patients who had stable disease, most of them had a reduction in their RECIST measurements for their target lesions, even if they did not meet criteria for RECIST response. So this was very impressive in a heavily pretreated population. And the second message for the study was that the duration of therapy was quite impressive. The median duration of time on treatment was 188 days, which is approximately 6 months. And if you put that in the context of many second-line chemotherapy trials in biliary tract cancers, where median PFS is 3 months, this demonstrates that not only in terms of response rates, but also durability of treatment, that this is a very exciting compound. So of course, if patients are on treatment for longer, we need to be concerned specifically about the side effects that may cause deterioration in quality of life. And overall, BJG398 was very safe and well tolerated in this population. Those toxicities are class effects of FGFR inhibitors. For example, hyperphosphatemia is an on-target effect of FGFR inhibition in the kidneys. So dose reductions and interruptions were rare, and only 2 patients out of 47 on the study needed to stop for adverse events. So taken together, the response data, the duration of treatment, and the toxicity would imply that this would be a very nice compound to develop further in FGFR2 fusion-positive cholangiocarcinoma. Now, there is another FGFR inhibitor which has presented data this year, gone by, in FGFR fusion-positive cholangiocarcinoma, and that's ARQ-087. The structure of that's shown on the left. Now, it's also a very potent inhibitor of FGFR1 to 3, but does inhibit other kinases at relatively low concentrations, and so we might expect the toxicity profile to be a little different. Preclinically, on the left, bottom left, it does also reduce FGFR signaling in FGFR amplified cell lines, and in mice models reduces tumor growth of FGFR2 amplified tumors in a dose-dependent manner. So again, strong preclinical rationale to proceed clinical trials. So the early results for ARQ-087 were presented at ESMO GI in Barcelona in the summer. And what we saw was they treated 21 patients with cholangiocarcinoma. 14 of these had FGFR2 fusions. And of that 14, 3 responded for a response rate of 25%. So it's comparable to BJG398. Because it was very early in the drug's development, no survival data were presented. And so what does this mean for patients? So these images are taken from the ARQ-087 poster at ESMO GI. And we can see here 2 patients who derived a benefit from ARQ-087. The first, previously treated with 4 other treatment regimens, had a nice reduction in size of liver metastases and lymph node metastases, and was on treatment for 10 cycles. The second, a reduction of 44% in liver metastases with 4 cycles and treatment ongoing at the time of data lock. So these suggest that the results for BJG398 and ARQ-087 demonstrate that targeting FGFR2 translocated cholangiocarcinoma has been therapeutically quite successful. So taking a step back, clearly there's no randomized data yet to support this treatment approach in cholangiocarcinoma. But if we go back to the results that Dr. Jane presented at ASCO in the summer, those 95 patients who had abnormalities in FGFR signaling, and 36 of these were treated with FGFR-specific targeted therapy. And there was a diversity in those, the BJG398, ponatinib, TAS120, and davitinib. So if we look at the survival for the patients who were FGFR dysregulated, who were treated with FGFR-directed therapy or not, those who were treated with FGFR-directed therapy had a better median survival at 44 months than those who were not. Now, there's a couple of potential biases in that analysis, but it does seem to suggest that for patients who have FGFR dysregulated tumours, that a therapeutic approach targeting the FGFR pathway may be better than a non-targeted approach. So finally, I just wanted to comment whether we have learned anything from targeting FGFR in other cancers. So in the study I'm running in London, the first 9 patients with FGFR2 amplified gastric cancer we treated with AZD4547. 3 of them had a really great response, and the others did not respond. So we really wanted to evaluate why that might be. And we had a very close look at their tumours using a digital pathology platform. And what we found was that even though patients were required to have FGFR2 amplification to participate in the study, the proportion of cells that had FGFR2, the proportion of tumour that had FGFR2 amplification was very variable. So it ran from 14% to 100%. And only patients with very high level and homogeneous FGFR2 amplification benefited from treatment and had a response. So this really suggested to us that heterogeneity of FGFR2 amplification was responsible for the differential responses that we saw to AZD4547 in that study. And as heterogeneity in cholangiocarcinoma has been discussed, assessment of the presence of the proportion of cells or the heterogeneity of FGFR2 fusion will be important in order to understand responses to these drugs moving forward. The second lesson that we learned from our FGFR inhibitor study was that the patients who responded to AZD4547, who had the highly and homogeneously amplified tumours, also had the amplification detected in the blood very easily using ddPCR, and no non-responding patient did not. Now, whether cfDNA tests for FGFR fusions I believe would be technically a little bit more challenging than detecting an amplification, but these, at least for us, the amplification, detecting the amplification in the blood was a better predictor of response than FISH in the tissue, and allowed us to circumvent that heterogeneity. So my conclusions would be that the FGFR signaling pathway plays a key role in cholangiocarcinoma. There's compelling preclinical evidence that suggests that FGFR fusion-positive tumours are addicted to FGFR signaling, and very encouraging data from Phase II clinical trials supporting FGFR-directed therapy in this patient population. And you know, it's only been 3 to 4 years since the first paper describing FGFR fusions in cholangiocarcinoma, and to be at this point with a positive Phase II trial is just really a testament to the cholangiocarcinoma community, investigators, and patients. And I would like to say well done, because I think that's a great achievement. Of course, there are always further questions, I think we need to do a lot of work to evaluate whether all the FGFR2 fusions which have been described are equal in oncogenic potential and druggability, what the interaction between FGFR2 fusions and other coexisting genetic events might be, the impact of tumor heterogeneity, which I've just discussed. And I think in particular, because we're seeing the emergence of these very active drugs in cholangiocarcinoma, and because resistance to tyrosine kinase inhibitors has tended to emerge very quickly in other diseases, that evaluation of resistance mechanisms will be absolutely essential. And I think actually that would be the next talk. So thank you very much.